Why can't we cure cancer? For decades, this has been science's holy grail, but despite billions of dollars and untold hours of research, the headline never arrives. In fact, most people don't realize that the majority of cancer deaths, some estimates say 80 to 90%, occur not because the disease is untreatable, but because tumors adapt and become resistant to therapy. If that statistic makes you uneasy, it should. It suggests that the problem isn't a single villain waiting to be slain, but an evolving ecosystem that outwits us at every turn. Why do enormous animals like elephants or whales rarely get cancer, while much smaller creatures like ferrets are riddled with tumors? Elephants have 20 copies of the tumor suppressor gene, TP53, and seem almost immune, whereas ferrets and opossums are among the vertebrates that get the most tumors. Meanwhile, a species called the naked mole rat produces incredibly long chains of hyaluronic acid in its tissues, essentially a biological jelly that stops cancer before it starts. Nature is full of paradoxes that tease us with answers. In this episode, we're going to explore why curing cancer is so hard. We'll venture far beyond the usual sound bites into evolutionary biology, physics, philosophy, and cutting-edge medicine. We'll ask whether a cure is even the right way to frame the problem, and we'll use analogies and thought experiments to unravel hidden complexities. By the end, you may find yourself thinking about cancer and life itself in ways you never imagined. When most people imagine cancer, they picture a single disease. In reality, cancer is an umbrella term for more than 200 different diseases. Each type has its own subtypes, genetic mutations, microenvironments, and behaviors. Even within a single tumor, the cells can be wildly different from each other. Some are aggressive, some are slow-growing, others lie dormant. This intratumoral heterogeneity means that a treatment may kill one population of cells while others survive and eventually repopulate the tumor. Think of a tumor not as a uniform blob, but as a vibrant city full of competing neighborhoods, each with its own rules and alliances. Our bodies are built from cells that cooperate through complex signaling networks. Cancer arises when a cell opts out of that cooperative contract, choosing to proliferate endlessly. That defection has many possible causes: mutations in oncogenes, loss of tumor suppressors, changes in the epigenome, and each combination creates a different cancer. One review likens the tumor microenvironment to an ecosystem involving cancer cells, immune cells, fibroblasts, endothelial cells, and extracellular matrix. These components communicate through chemical signals and mechanical forces. The microenvironment supports tumor growth by adjusting metabolism, switching to aerobic glycolysis or glutamine metabolism, and by reprogramming immune cells to become allies rather than foes. It even establishes pre-metastatic niches in distant organs using immune cells as diplomats. Because of this diversity, there will never be a single cure for cancer. Every tumor is like a fingerprint. When doctors design a treatment plan, they must consider not just the cancer type, but the subtype, the stage, the patient's genetics and comorbidities, and even the patient's preferences. It's like designing a tailored suit. One size doesn't fit all. The biologist Douglas Hanahan famously described cancer as having hallmarks, distinctive capabilities that normal cells lack. These include sustaining proliferative signaling, evading growth suppressors, resisting cell death, enabling replicative immortality, inducing angiogenesis, and activating invasion and metastasis. More recently, researchers have added new hallmarks, reprogramming metabolism and avoiding immune destruction. Enabling traits such as genome instability and tumor-promoting inflammation fuel these hallmarks. Emerging concepts like phenotypic plasticity, epigenetic reprogramming, microbiome interactions, and senescent cells further complicate the picture. Each hallmark is a challenge for therapy. Together, they make cancer a moving target. Most cancer deaths aren't caused by the primary tumor. They result from metastasis, the process by which cancer cells spread to other organs. It is estimated that over 90% of cancer fatalities are due to metastatic disease. Metastasis is not random. Colon cancers tend to colonize the liver and lungs. Prostate cancer favors bones. And these patterns hint at chemical crosstalk between tumor cells and distant tissues. The ability to metastasize is itself a form of evolution. Only cells that acquire the right combination of traits, motility, invasiveness, immune evasion, can succeed. On top of metastasis sits the problem of treatment resistance. When we expose a tumor to chemotherapy, targeted therapy, or radiation, most cells die. But a few survive because of pre-existing or newly acquired mutations. Like weeds that resist herbicide, those survivors repopulate the tumor with a vengeance. This dynamic is why resistance contributes to 80 to 90% of cancer deaths. Scientists are now experimenting with forward engineering tumors, intentionally inserting genes into cancer cells that both make them resistant to a drug and cause them to produce a toxin, so that when the resistant cells take over, they commit suicide and kill their neighbors. It's a mind-bending strategy, using evolution against itself, but early results in mice show promise. Our immune system is constantly patrolling for rogue cells, yet tumors often grow unchecked. One recent study found that early colon cancer cells switch on a gene called SOX17, which downregulates interferon gamma signaling and reduces antigen presentation. By turning themselves invisible, these precancerous cells avoid being attacked. This is just one of many tricks cancers use. They recruit regulatory T cells to suppress immune responses, express checkpoint proteins like PD-L1 to exhaust cytotoxic T cells, and even reshape their surrounding matrix to create physical barriers. Immunotherapies, checkpoint inhibitors, CAR T cells, and cancer vaccines represent a revolution in oncology, yet they also illustrate the complexity of the task. Antibody-drug conjugates, ADCs, deliver toxic payloads directly to tumor cells, but finding targets that aren't also on healthy tissues is challenging. Cancer vaccines aim to teach the immune system to recognize tumor-specific mutations. Trials are underway for cancers with both high and low mutation burdens, and researchers are debating whether vaccines must be personalized for each patient. Meanwhile, cell-based therapies such as CAR T cells work spectacularly in some blood cancers, but struggle against solid tumors. Efforts to develop off-the-shelf versions and Boolean logic gates to increase specificity are ongoing. Despite these advances, rare drug-tolerant cells and cancer stem cells can survive and later regenerate tumors. The possibility of a cure is highest when cancer is caught at its earliest stage, when it can be removed surgically or targeted with minimal disease burden. Regular screening saves lives. Mammography has driven breast cancer survival rates from 75% to 91%, largely because of early detection and advances in hormonal therapy. Yet not all cancers have obvious symptoms or available screening tests. Uterine cancer, for example, is common, but lacks a standard screening exam. If symptoms are subtle, the cancer has time to progress to later stages before it is detected. Even when screening exists, socioeconomic barriers like transportation, time off work, and insurance can prevent people from accessing care. Doctors have a saying, "Stage is the most important prognostic factor." For colon and rectal cancers, the 5-year survival rate exceeds 90% for stage 1 disease, but falls to around 12 to 16% for stage I4. For lung cancer, 5-year survival is 67% when caught at stage I, but only 6% at stage I4. These stark contrasts show that while a universal cure may be elusive, early detection is our most powerful tool. The quest for a cure isn't confined to human biology. Peto's paradox observes that large animals with more cells should get more cancer simply because there are more opportunities for mutations, yet they don't. A study spanning 292 vertebrate species found that everything gets cancer, but the prevalence varies wildly. Black-footed penguins and common porpoises get very few tumors, while ferrets and opossums get many. The study also hinted that animals with longer gestation times, and often larger bodies, invest more in somatic maintenance, meaning they're better at repairing DNA damage and preventing mutations. Elephants illustrate this principle. They carry around about 20 copies of the tumor suppressor gene TP53, which triggers cell death when DNA damage occurs. Then there's the naked mole rat. These wrinkled, nearly blind rodents live for more than 30 years, 10 times longer than mice, and have an astonishingly low cancer incidence. Researchers discovered that the naked mole rat's cells produce high molecular weight hyaluronic acid, chains 6 to 12 million Daltons long. that accumulate in the extracellular matrix and activate a cell cycle inhibitor called P16. This triggers early contact inhibition, meaning that when cells touch each other, they stop dividing. It's as if the tissue enforces strict social distancing, preventing any one cell from gaining a competitive edge. When scientists introduced the gene responsible for this giant hyaluronan into mice, the mice displayed increased cancer resistance, hinting that molecules like hyaluronan could inspire future therapies. These examples underscore a profound truth. Evolution has already experimented with hundreds of anti-cancer strategies. Some species duplicate tumor suppressor genes. Others slow down metabolism or produce unique extracellular matrices. Studying these mechanisms offers fresh perspectives beyond traditional drug development. So why can't we cure cancer? The simplest answer is that cancer is not a single enemy, and it isn't just a disease. It's an inevitable byproduct of multicellularity. Wherever cells divide and genomes mutate, the possibility of a renegade clone emerges. Heterogeneity, microenvironmental interactions, immune evasion, and evolutionary dynamics make the battlefield uneven and ever-changing. But there's another reason. The idea of a cure implies finality, an end state where the threat is gone. Biological systems rarely offer such certainties. Even if we eliminate every detectable cancer cell, dormant cells can hide, or new ones can arise later. Instead, many scientists now talk about managing cancer as a chronic condition. Just as HIV has been transformed from a death sentence into a manageable disease thanks to antiretroviral therapy, certain cancers, like chronic myelogenous leukemia, are controlled for decades by targeted drugs. More than half of patients diagnosed with cancer now survive longer than 10 years, a dramatic improvement over previous generations. The overall cancer death rate is steadily declining, and millions of survivors live full lives. New technologies are accelerating our understanding. Single-cell sequencing and spatial omics allow scientists to map the genetic and epigenetic diversity within tumors and identify rare cells that drive recurrence. Artificial intelligence is being used to predict how tumors will evolve and which drug combinations might preempt resistance. Gene editing tools like CRISPR are being explored to correct mutations or engineer immune cells, and the concept of combination therapies, inspired by the multiple hallmarks of cancer, is becoming mainstream. By targeting several pathways simultaneously, we may slow evolution enough to keep tumors under control. However, progress is not just about technology. Health equity and clinical trial representation matter deeply. The Mayo Clinic notes that the lack of representation from diverse populations has hindered progress, and that barriers like transportation and work obligations prevent people from accessing care. The disparities in survival rates between racial groups, such as Black and white women with breast cancer, remind us that social determinants of health are as important as genetic determinants. A future cure must address these systemic issues too. At this point, you might feel that the answer to "why can't we cure cancer?" is disheartening. But here's the twist. Progress lies in embracing complexity rather than seeking oversimplified solutions. Cancer is a testament to the creativity of evolution. Each tumor is a miniature world, and each species has its own story of resistance or susceptibility. We're learning from elephants, naked mole rats, and penguins. We're redirecting tumor evolution. We're personalizing therapies. We're catching cancer earlier and improving survival. Perhaps the hardest problem in medicine is really a mirror reflecting our own nature. Cancer forces us to confront the very mechanisms that allow life to flourish: growth, diversity, and change. Instead of searching for a magic bullet, we can aim for a world where cancer is detected early, controlled effectively, and, in many cases, prevented. And maybe, just maybe, by understanding why enormous creatures rarely get cancer and how tiny rodents resist it, we'll unlock insights that reshape our relationship with disease. So the next time you hear someone ask, "Why haven't we cured cancer yet?" you can answer with a smile, because cancer is not one disease, but a complex tapestry woven into the fabric of life.